Yeah. Good morning, good afternoon, good evening. My name is Simon Agwale, Chair of AVMI Vaccine Technical Working Group. It is my pleasure to introduce our speaker, Mr. Mark Emalfarb, who will be talking to us about making healthcare accessible to patients globally through the use of an engineered eukaryotic filamentous fungal cell lines, the C1. Mr. Emalfarb is the founder and CEO of Dyadic International Incorporated, which celebrated its 41st anniversary in January 2020. Dyadic is a NASDAQ-listed global biotechnology company with operations in the US and EU. He is the inventor of more than 25 US and foreign biotechnology patents and patent applications. Mr. Emalfarb has led Dyadic through a variety of evolutionary changes, including its recent transition from industrial biotech company with the sale of its industrial biotechnology business to DuPont for $75 million in cash. This repositioned Dyadic into the biopharmaceutical industry, where the company is leveraging its industrially proven, patented, proprietary C1 gene expression platform to help enable and speed the development, lower the cost of biologic, and improve the performance of biologic vaccines and drugs. Since the DuPont transaction, Dyadic has entered into more than a dozen research collaborations with big pharma, biotech, academic, and government organizations, such as the US Frederick National Laboratory, to engineer its patented and proprietary C1 cell lines to produce a number of COVID-19 vaccine candidates, which will be utilized by the Vaccine Research Center of the National Institute for Health. Other institutions that Dyadic is collaborating with include the Sanofi Aventis, the Serum Institute of India, and several others. So over to you, Mr. Emalfarb, who will be talking for about 40 minutes, and then we'll have 15-20 minutes for question and answer section. Over to you.
Great, thank you, Simon. First and foremost, I want to thank everybody for attending today's presentation. As the title points out here, there's an urgent unmet need for affordable, not just halal, but vaccines in general. Of course, in Africa, there's a major portion of the population that would like a halal vaccine. And we made enzymes for halal for over 20 some years with the same C1 cell technology. So the point here is that we believe we have the ability to not only help Africa become independent in terms of its own needs, but also to basically work on a pandemic and or just traditional vaccines and drugs quicker, faster, and more affordably using very simple manufacturing capabilities. So I think the point that we're trying to do here for the African continent has become, will they be dependent on others, or will they be able to take care of their own needs? And I think this technology, because it's very easy to tech transfer, you use standard E. coli fermenters, it can produce large amounts or massive quantities of antigens or therapeutic drugs and proteins quickly at flexible commercial scales at very, very low cost using raw materials that are locally available. So I think this will be a godsend for the African continent and as well as the world in general. And we're here to help make that happen. So one of the challenges, of course, in addition to making a vaccine, is in this particular case, making a halal vaccine using the raw materials and the ingredients that allow you to propagate, isolate, purify, and formulate a vaccine that would be halal certified. And again, since we made enzymes for over 2 decades that were both kosher and halal, we believe that the C1 technology platform and the raw materials used will be certifiable for the halal vaccine as well for Africa, which has a large population of Muslims in it. So one of the problems that we're all facing today, whether it's just the first break of COVID-19, a variant of concern, which makes it even more difficult, is a vaccine that's unavailable or unaffordable is 100% ineffective. And although America and Europe and Israel and some of the other countries have access to vaccines, most of the world is still struggling. The variants of concern are growing, they're emerging, and we think that this technology could have prevented a lot of those variants of concern from emerging in the first place. And we think that we can provide not only the capabilities, but the technology for today, tomorrow, and the future pandemics and just traditional vaccines and drugs that can make it more affordable and accessible to not only all the population of all the different countries in Africa, but also to allow the African subcontinent to generate income and revenue because of the extra capacity and the strength and the productivity to basically sell products and generate revenue in addition to taking care of their own needs. The technology is based on a filamentous fungal cell that we spent over 25 years engineering, but first random mutagenesis, then to synthetic biology, genetic engineering. This cell has very unique properties. It doesn't grow like a typical filamentous fungi, and don't confuse it with yeast, because filamentous fungi secrete significantly higher levels of proteins. And this particular fungal cell not only secretes significantly higher levels, 5 or 10-fold potentially than a yeast cell would, well, you'll see several hundred times potentially greater than an insect cell like baculovirus. It does so at high levels, flexible commercial scales, high purity, and very, very low viscosity with unique glycan structures that are very human-like, much more human structures from the C1 cell line. And we've also now glyco-engineered C1 cells to have human-like structures as well. So the goal here is when you want to produce a antigen or a protein, you want to do so at high purities, preferably without viruses or endotoxins. Of course, E. coli has endotoxins you have to remove. We don't have endotoxins in C1 to remove in the first place, and we don't have viruses like CHO cells. It's robust, it's versatile growth conditions. We'll get into the temperature and pH range, and a low viscosity unique morphology is an advantage. And the scales that you can grow from a microtiter plate up to massive scales of 500,000 liters. We could rapidly develop new cell lines as variants emerge. Stable cell lines you can produce very high levels at very low cost. They can be tech transferred, and then high yields, low cost production to reduce the manufacturing footprint. So some of the benefits here to the C1 cell line are its scalability and ability to grow at multiple scales, from small fermenters to large fermenters and everywhere in between, where you can actually produce more for less. Even in a small volume footprint, you can produce tens of millions, if not hundreds of millions of doses. And with a little larger footprint, you can produce billions of doses, potentially on a monthly basis. So it's very productive and very low cost. One of the advantages of the technology that we've developed is we don't do random mutagenesis, we do site-specific mutagenesis, where we take a gene sequence, either 1 or 2 copies, and you can see here with the green original gene, you can put 1 or 2 copies in, and within roughly 60 days or less, you have a stable cell line that can produce quantities that go into animal studies and be ready to go move towards cGMP grade, because we're not going through transient cells, we're producing stable cell lines from day 1. And if you want to change the sequence from, for example, the Wuhan gene to a South African variant, or Brazilian variant, or UK variant, or now the Delta variant, you just pop out the gene and pop in the new gene, and all of a sudden, within 2 months or less, you have a stable cell line producing high volumes of the new variants of concern. One of the big advantages of the technology is it grows in single-use or stainless steel fermenters. In this case, we're showing you stainless steel fermenters from pH 5 to 8, temperatures of 20 to 45 degrees Celsius. And you can use a standard E. coli fermenter that's virtually available in most countries around the world. You can go up to 500,000 liters. If you ran at that scale, you can certainly solve the quantities of antigens probably for the entire world in a month, but you don't need to run at that large scale and still pump out billions of doses or hundreds of millions of doses on a monthly basis. And we run on very low cost media glucose, and a few amino acids, all cGMP chemically defined media. You can also grow in a single-use bioreactor, and this is an example of an XBR50, where we ran, in this particular case, it was an antibiotic fragment in stainless steel and in single-use reactors, and ended up with virtually the same productivity levels. This is a flow chart of how simple and easy it is. Once you've generated that stable cell line, going from the freezer to the end of fermentation batch, using C1 cells, it's only 12 to 14 days, roughly 5 days in the fermenter, versus 41 to 54 days of a CHO cell. So you can run 3 batches at higher levels at lower cost of the same product in much lower cost media. So you can make more for less and produce billions of doses potentially of antigens quickly, rapidly, and affordably. We spent 5 years getting prepared for the pandemic in the European Union in a cooperation with the called Zoonosis Anticipation Preparative Initiative. This is one of 2 governmental programs we were working on to get ready to combat pandemics, epidemics, and potential biological threats. And we were very successful. You can go online and click on any links below, and those are reviews of a 2-day, 8 hours each day, reviewing the 5-year programs. But the summary here, as mentioned by Dr. Audene, the coordinator for the ZAPI program, is the C1 technology far exceeded their expectations, and we'll get into how and why that happened, the data that demonstrates that. So this particular slide shows you Dr. Albert Osterhaus, which is one of the coordinators and the participants in the ZAPI program, where he mentioned that if nothing else came out of this 5 years of research and development and getting prepared for zoonosis disease to be able to quickly react to the problem, C1 would be an ideal spinoff. If nothing else happened good, that was good enough. And then the next gentleman here you can see is Dr. Alexander Brix from Boehringer Ingelheim, and he talks about the quantities that you can produce at small scale, the need not to have to produce at large scale, a smaller footprint, which allows you to have multiple sites, or if you want just one single site to produce the billions of doses that you want quickly and affordably. An example of the C1 cell line you saw earlier versus the CHO cells, 3 to 4 batches versus 1 batch in the same time at a lower cost at higher yields. This is about a comparison to baculovirus cells or insect cells. And in that program, the SPV antigen and the RVFV antigens were used as proofs of concepts. And if you can see here, we were able to produce the SPV antigen at 1.8 grams per liter, the RVFV at 1.24 grams per liter. We're now at about 1.7 or 1.8 grams per liter of that. And if you look at that compared to the insect cells or baculovirus cells, it was produced at 300 times the level. So whether it's 50 or 100 or 200 or 300 times level, it's a massive difference. You can make significantly larger quantities and much lower cost vaccines, and you have in less time. You can see we did it in 121 hours, and it took them 192 hours to produce far less. So in addition to the European Union, as I mentioned, we worked with 2 governmental programs before the pandemic. EU starting in 2015, 5 years ahead, 5-year program. Here with the Israel Institute for Biological Research, we did the same thing, getting prepared for pandemics and epidemics and potential biological threats. First thing we did is we made an FC fusion enzyme to help provide them as a countermeasure against nerve agents or sarin gas. But related to this point, they, as well as the Europeans, chose to use C1 to develop what they call as a simple, fast, efficient way to neutralize the virus. And you'll see coming up, we were very effective in the mice studies, which had to generate excellent immunogenicity responses with high titers of neutralizing antibodies. And we carried out with the IIBR 2 successful challenge studies for transgenic human ACE2 mice very successfully. So the approach here was keep it simple, make more than less, keep it safer. So with SARS and MERS and COVID in the past, the full spike protein had side effects that were detrimental. They were making antibodies that you didn't really necessarily need. In the case of COVID-19, if you can just neutralize the virus, and there's more and more publications coming out that the RBD alone has the potential to neutralize the virus effectively and efficiently. And the advantages of using the RBD versus the full spike are it's a smaller protein, it can be made in larger quantities at lower cost, and you can avoid the potential issues of ADE or ERD, enhanced respiratory disease, or antibody-dependent enhancement that you saw earlier with COVID and SARS and MERS. And so 80% or approximately of the neutralizing antibodies come from the RBD. So it's a more focused neutralizing antibody antigen that's a higher concentration of the neutralizing antibodies. So in the study here with the mice, first we did in vitro biomolecular binding kinetic assays and showed that the CHO-produced RBD was virtually identical in performance to the C1 produced RBD. And as we mentioned, we went to the mice trials and showed that the neutralizing antibodies bind to all the RBD epitopes and demonstrated high immune response and protection against SARS-CoV-2 virus. And then recently, successfully, the two ACE2 hamster studies, transgenic mice studies, which we'll get into coming up. So this shows you here that in the challenge mice studies, we did a 2-dose and a 3-dose regimen. And the reason we did a 2-dose and a 3-dose regimen was because whether you were going to need boosts on a continuum basis or not. And if we do so, we think the RBD as a focused neutralizing antibody vaccine may be a tremendous booster vaccine to the DNA vaccines. And as you know, today, there's a heterologous sort of boost technology that they're testing where you can take the AstraZeneca, or you can take the Pfizer, the Moderna, the J&J shots, and boost the activities of neutralizing antibodies. And in particular, if you use a protein or recombinant protein vaccine that's targeting higher concentrations and higher numbers of neutralizing antibodies, we expect that that would be a potentially even better, safer boost than using the full spike over and over again. And you see here in the first trial with the two shots, prime and boost, was very successful. And on the here, you can see the same thing with the three shots, how much higher the antibodies came when you had the booster added. They went up exponentially high. And so these are challenge studies with ACE2 transgenic mice, either live or die. In this particular case, in the 2 shots, 7 out of the 8 mice lived. The 1 mice didn't have the neutralizing activities. As you know, mice are variable. And then in the 3 shots, you see that all of them lived and the neutralizing antibodies were extremely high. So both of those are successful. And this is, by the way, keeping it simple, using L-hydrogel or alum with 20 units per gram or 20 micrograms with a very affordable, low cost, readily available adjuvant. And we used alum in this case because we needed to make billions of doses potentially for a global population. And as you know, some of these other adjuvants, people can't get access to them, they're running out of them, and that would affect it. So we kept it simple and at low cost and effective. And we're moving forward now with what we call our DYAI 100 vaccine in combination with the scientists from the Utrecht University, Erasmus Medical Center, TIHO, 3 of the top 20 coronavirus scientists were involved in the ZAPI program with us, supported by the Israeli government as IRBR, the development program that we just talked about, with ParaXL, CMC consultant and regulatory for CMC, Biotechnology General, which is a division of Ferring Pharmaceutical, is using the cGMP manufacturing and the standard E. coli fermenters. And we've already run two engineering batches and 1 cGMP batch. We have a tox study ongoing that looks very promising. They've already sacrificed all the rabbits. Everybody's looking healthy and happy. So we'll have those results coming up here, at least preliminary results coming up in July. And then the clinical trial is going to be overseen by CR2O as we move that forward. So one of the things I wanted to make clear, we're really using a technology platform, and we're offering this platform not just for our own candidate, the DYA 100 or the DYA 200, 300, or 400, if we actually make a tetravalent or a trivalent vaccine with the Wuhan South African, Brazilian, UK, potentially Indian or Delta variant. But we've also made the full spike. And we've made these things alone and with spike tags to make nanoparticle vaccines, conjugating them, as you see here on the bottom, with either MI3 or hepatitis B or other nanoparticles. We've already successfully done all that, and we'll talk about that in a publication that just came out last week. We've also made monoclonal antibodies. We've made, in addition to the full spike or the nanoparticle vaccines, we've also made the FCRBD, and of course, influenza, MERS, RVFV, SPV, et cetera. And you can see the high purity and the high productivity. So today there's virtually 4 different types of vaccines that are being produced. You've got the whole virus vaccines, you get the protein subunit vaccines, which is what we're making, nucleic acid vaccines, or the viral vector vaccines. We believe, and it is growing evidence, that number one, we potentially, based on our history of making enzymes with halal components, can manufacture a halal vaccine using the right raw materials that are halal approved. But also we think recombinant protein vaccines, not only can we make more of them for less using standard E. coli fermenters, but we think potentially they may provide even a better booster or maybe even a better protection on long-term than some of the DNA, and they may be safer. And they're more traditional. This is what you've been using for flu shots or for insulin shots for decades. And this is, I think, a potentially safer way to deliver a vaccine that's more traditional, that can maybe get some of the people that have hesitancy of taking a vaccine to actually take vaccines in a more traditional way. So as I mentioned, recently, June 15th, publication in Vaccine publication, a trade journal that's a highly respected trade journal. And again here, and you can go in and find that on our website, the C1 produced antigen, in this particular case, with the SPV antigen, provided excellent response, safety, efficacy, and protection with very, very high productivity and very, very low cost. It can be manufactured at flexible commercial scales. This is an example of, in that particular case, we made a nanoparticle vaccine. And here we show you with the RBD spike tag without a C tag, we were able to make 700 to 800 milligrams per liter in only 55 hours with very high purity. That's an enormous quantity of antigens in such a short period of time and a very high pure level. So as I mentioned, we can very quickly take one gene out, put another gene in without changing the genetics of the cell. From a regulatory standpoint, everything stays the same with the sequence. And within 2 months, we were able to generate the British variant, the South African variant, the UK variant at the same levels or very similar levels to the Wuhan. So it shows that the technology is flexible, robust, versatile, rapid production of stable cell lines that can produce very, very large quantities at very low cost. And we're now working on the Delta variant as well. And we're here and we can provide mixtures of all the different variants, monocomponents, trivalent, tetravalent vaccines, or the vaccines for the next generation where people are coming up with new gene sequences that have even potentially better performance and better protection. We should load them up into a more efficient cell line that can be produced large amounts at low cost using standard E. coli fermenters. And so here's an example. In our research program, we're able to reach up to 3 grams per liter on an RBD protein. And if you took 2 shots of a 20 microgram twice, in this particular case, and you used 80% of the volume of the fermenter and you recovered 70% of it, you could produce 2 shots for 1 billion people in 23,810 liters of a fermenter. So if you happen to have a 20,000 liter fermenter, you could produce enough vaccines or antigens for the vaccines in a week for 1 billion people. If you want to have 100 million vaccines produced and you use a 2,000 liter fermenter, you can do that in 5 to 7 days again. And so if you want 1 billion, you can run basically 10 different fermentation runs, and each run's somewhere between 5 to 7 days or less. And then if you want to go down even further, you can go into 200 liter fermenter and get about 10 million doses out of the antigens. And then if you want to make nanoparticle vaccines, you even need less fermentation capacity, for the antigen, but then you need the fermentation capacity in addition for the nanoparticles. So I think you can see here, compared to all other manufacturing platforms, we can produce far more faster, quicker, flexible commercial scales using standard E. coli fermenters and local raw materials. I was showing you there, but somehow there's a voice here, but this is another example of hemagglutinin or seasonal flu. This work was done by Sanofi Pasteur, and it shows that not only did we make larger quantities of lower cost hemagglutinin, but it was safe, effective, and protective in the mice trials. And this is one of the largest seasonal flu producers or vaccine producers, Sanofi, and it's the data that we can share. And there's a patent application and a publication out there on the web, and we'd be happy to supply that to you if you have an interest in seasonal flu. And we may combine the seasonal flu with a COVID shot as well as we move forward. In addition to obviously producing antigens, we produced monoclonal antibodies of all types and varieties, including a SARS-CoV-2 antibody. And in the case, as we showed you, we can produce 3 or 4 batches in the same time. You can produce 1 batch of a CHO cell, potentially at greater quantities, faster, lower cost, immediate cost, without the need for removing viruses. So you remove 2 viral inactivation steps. And then this is just an example. If this technology, unlike mRNA, not only can you produce an antigen, you can produce an antibody, you can produce a Fab, a trispecific, a bispecific, all kinds of opportunities. It's broader, more versatile, more robust, more scalable. So it can provide lower cost healthcare to the African continent for a variety of diseases, whether it be cancer, potentially insulin for diabetes, rheumatoid arthritis, in addition to all the different types of vaccines. So 2 examples, and similar to this is what we're trying to accomplish here now on the African continent. We're working with Meditox in South Korea and Asia, Southeast Asia. Meditox had worked with both CHO cells and insect cells. And as you can see here from Dr. Yang, who worked closely in the past with C1 and is now working on either a trivalent or tetravalent or a monovalent COVID-19 variant of interest or variant of concern, that they believe that the C1 technology is the most realistic one to use to make large volumes of low-cost proteins. It can provide the quantities at the cost the world can afford. And then we're working on a similar program in India with Syngene, which is a subsidiary of Biocon. And again, they're evaluating it not only for COVID-19, but as a differentiated vaccine platform and drug platform using C1 cell lines to bring affordable healthcare for a variety of different product opportunities for their population and beyond. And again, we're trying to now work with you in Africa to come up with a simple, robust, easy way to develop and produce your own vaccines or antibodies or drugs in the continent. Simplicity, simple manufacturing, robust, quality, easily to deploy, high productivity. And it's really important to consider it's not sustainable to be paying the price for the massive amount of doses that you need on some of the vaccines that are being sold today. I mean, some of these people are selling $20 billion. There's no way that the world can afford the price tag going forward. And certainly in Africa, you need not only high doses, you need it at reasonable cost that you can afford to provide it without putting yourselves into extreme debt. And so the infrastructure, the concept, location, the question really is, do you build one large facility in Africa, South Africa, to take care of the entire continent, or do you build multiple facilities and smaller size footprints, and you're still able to, as you remember, pump out hundreds of millions, if not billions of doses with even smaller fermenters? And really, the total investment that we're talking about here is de minimis compared to the disastrous societal and economic damages, and worse yet, the lives being lost because we weren't ready, or well, we were, with the zoonosis anticipation ZAPI program and the Israeli program. The point here is we're here to help, and together we need to find a way to put the infrastructure on the continent in a way that makes sense, not just for COVID-19, but for other drugs and vaccines, of traditional drugs and vaccines, and to be better prepared for the future when we need it. And the message is bringing global health access, affordability, and equity. And I think together we can accomplish that goal. And we can do so, I think, in a much more efficient way using the C1 platform technology than any other platform technology that's available today. And I want to thank you for taking the time.
Well. Thank you very much, Mark, for this wonderful presentation. Please, if you do have any question, type it in the chat room. We'll read it out for Mark to respond. I have a quick question. The challenge experiment that you did in mice, did you challenge the mice with all the variants of concern or just with one variant?
Well, in that experiment, we just did it with one variant. It was the Wuhan variant. And as you noticed, we used very, very high levels, like 2000 PFU to challenge them. But there is work ongoing now with some of the other collaborations we have ongoing where we're seeing some effectiveness against the different variants.
Okay, great. I have a question here for you in the chat room. Say, please, can you confirm the main titer differences from C1 versus CHO on some typical monoclonal antibodies you have seen?
Yeah, so let me go back to a slide that can sort of highlight that. So using the non-glyco C1 cell line, we've actually produced a MAB at up to 24.5 grams per liter or 3.5 grams per liter per day. Number one, consider the fact that we're running at half the time in a fermenter, or 1/3 and 1/4 the time when you go from freezer to batch. But in a fermenter alone, you could just double that because you're running 12 to 14 days in a fermenter in a CHO cell. So I think the average CHO cell would be around 4 grams per liter. And so if you did this 4 grams per liter and you do that in 12 to 14 days, in this case, you got 3.5 grams per liter per day, you can see that there's a potential. And this is not even optimized, by the way. This is just popping the gene heavy and light chain into one spot, one copy of each gene, site specific, and we came up with that result. Now, variety of different CHO cell or different sequences are coming out with different productivity levels. But people have worked with CHO for 35 years, and we're just getting going and already getting enormous quantity advantages in cost. And the media cost is a fraction of the cost because we use glucose and amino acids. So no viruses, so there's a lot of benefits. And we just go through protein A and we end up virtually with the same purity that you would get out of a CHO after a protein A to go on to downstream processing.
Great. There's another question here. Somebody is asking when the C1 COVID vaccine will be in human trials.
We are working on getting that done sometime started by the end of this year is our goal and objective.
Okay. And I think some African manufacturers or countries will be interested in this platform. Can you briefly take us through the tech transfer procedures?
Yeah, it's relatively simple. By the way, we've tech transferred it all over the world for decades. On the industrial side, we tech transferred it to companies like BASF, Shell Oil, Abengoa Bioenergy, DuPont. Already on the pharmaceutical side, we've tech transferred it to Meditox, Epigen in India. Now we're working on Syngene with the Biocon in India, and we've tech transferred it to the Israelis. So it's very simple. It's a microbial fermentation. The unique morphology of C1 allows you to grow it under low viscosity at very high yields. So it's a simple tech transfer. So we would just be able to provide the cells. Typically, we engineer the cells with a gene of interest. So, if you go back to the slide here, depending on what gene you might want to put in, if you want to put in, well, we already have a South African cell line, but depending if you want to say the Delta variant, the new variant, once it's available in a month or two, you could just transfer it, or the Brazilian in Wuhan today, or the UK. very rapidly, very quickly. But if you have a new gene sequence for a whole new vaccine, again, we can go back to the slide that we started off at the beginning. We can very quickly just take the gene sequence without changing the cell line, pop out the gene already that exists, put in a new one, and within 60 days or so, you now have a whole stable cell line, not like CHO, it's not a CHO pool, it's a stable cell line with 1 or 2 copies, producing high levels of protein very quickly. So the tech transfer is easy. If you can grow yeast or you can grow E. coli in a microbial fermenter, you should have no problem mastering this very quickly.
Great. I have another question here for you. Can your system accommodate investigating polyvalent vaccines?
I didn't understand it. Can I do
It
what?
says, can your system accommodate investigating polyvalent vaccines?
I'm probably not the right guy to answer that question.
Okay. And of course, I would guess yes, because you are able to put all the RBDs in one construct, right? So
Right.
the answer will be yes. Now, I have another question here. It seems that C1 is a best-in-class quality platform that makes safe and effective vaccines. The next step is to tech transfer to Africa to a CGMP facility. Can Mark comment on what will be the next steps in order to tech transfer, and how soon could a vaccine study be set up in Africa?
Okay, so the tech transfer could start immediately, okay? And we've already taken it through the engineering batches in the first CGMP batch. So we have all the protocols in place. The question is, what party in Africa, where it's located, what your CGMP capabilities are, what size fermenter you have. Remember, we're scaling down, not scaling up. We've actually run at 500,000 liters in the industrial side for the biofuel enzymes, 150,000 liters, 60,000 liters. So whatever you're going to run at, it's going to be much smaller than what we've done for over 2 decades. So, we're here to support you and to do things as quickly as possible, but however, as accurately as possible and as safely as possible. But we can do it starting tomorrow if we have the right infrastructure in place in Africa with the right party. And the beautiful thing is you can use glucose or you can use sucrose. So the raw materials are readily available in the continent.
Okay, I know there are various ways you can tech transfer. Do you require upfront payment commitment from the manufacturers, or how is it done?
Well, number one, we're flexible. So if you're a large pharma company, we would like to get an upfront cash, milestones and royalties. If you're a small biotech, we can work on a more flexible arrangement. Our goal and objective is to put shots into people's arms to get safe, protective shots into not just in Africa, but on a global basis. So again, depending on the party, the resources, it could be an equity position. So we're flexible and entrepreneurial. So we can move quickly, we move swiftly with great flexibility. So we have certain companies where we take an equity position in and we get milestones and royalties. Other companies, you get upfront cash, milestones and royalties without equity. So we're flexible. Depends on the party and what they bring to the table. It's a win-win relationship.
Okay, great. So I guess manufacturers that may be interested in this platform are free to contact the AVMI Secretariat so that the Secretariat will put them directly in contact with you so that you can work out your bilateral agreements, right?
Yes.
Great. Well, any other question? We are waiting for your questions. You can go to the chat room, type your question, and then we'll read them out. We still have some time. Yeah.
Yeah, well, while we're waiting, if we, Simon, we could just share the vision. The vision here is to enable the African continent and the different countries within Africa to not only develop COVID-19 vaccine, but just as importantly, to use this technology to make sure that whether it be malaria or be HIV or a variety of other vaccines, to really take care of the continent in an affordable way that everybody can get access to healthcare, okay? And I think that's a dream and a vision people have had. And I think together, using this highly productive cell line that's been developed over 2.5 decades and proven in the industrial world to be scalable, productive, robust, and versatile, to make things at high volumes at low cost, is we have a chance to really accomplish what the world needs, and that's health equity. And together we can do that. And so you have lots of universities developing all kinds of new vaccines and antigens and all kinds of potential therapeutics. And together we can try to make those more affordably in larger volumes so that we can get them into a greater portion of the population.
Great. I have another question here for you. Any issues to note with respect to specific raw material supply during pandemics?
No, I mean, to be honest with you, and I'll go back, well, it's this screen right here. When we designed this, we could have gone with what you call a nanoparticle vaccine, which you see here, which is what we did. And I suggest if you get a chance, you can ask for this paper and publication, but it's also on our website, dyadic.com, Media Center under Scientific Publications. What we did is we didn't use in this particular case, the nanoparticle vaccine because C1 is so productive. The nanoparticle component would have been let's say, bottleneck. You can make hundreds of millions of doses, but you couldn't make billions of doses rapidly, quickly. But because we can produce massive amounts in comparison to, let's just say, insect cells or baculovirus, as you can see here, forget whether it's 300 times better or 50 times better, or 100 times more productive, we have the ability and capability to use higher doses of the RBD. So we're using a higher dose antigen because we have the luxury of doing it, which you don't have in a baculovirus, which is why companies like Novavax and others are using nanoparticles so they can stretch the doses out. We have the luxury of not having to do that. So we can make a nanoparticle vaccine or a non-nanoparticle vaccine, depending on what you want and the effect and the performance, but at a higher dose with just alum. And again, as I mentioned, this data is not with these liposomic adjuvants that are maybe effective, more effective, but this is survival and significant success just with alum, getting high neutralizing antibody titers. And alum, just to give an example, we've gone to people and said, hey, can we get whether it's AS01 or MF59, and they're not available. So we used alum because when you want to get it's proven, it's tested for decades, and guess what? It's pennies on a shot instead of dollars on a shot. So we want to make a vaccine that the world can have access to and afford, and not run out of some components. So as far as we know, there's no issues. We don't need plastic bags for single-use bioreactors. We don't need fancy adjuvants, which drive up the cost. We made it simple. It's RBD, alum, and virtually nothing else in there. And to produce it, it's glucose with a few amino acids, maybe some yeast extract. So most, if not all of these, are readily available on your own continent.
Great. So in terms of, there's another question here, in terms of clinical plan, where will the clinical trials be performed?
Well, currently we're targeting Germany for the first phase 1 clinical trial. So that's the first step. And we'll see when we get there if there's enough people that haven't been vaccinated yet to actually do that. And alternatively, we are in discussions with people to potentially run it in Africa and other countries as well.
Okay, great. So have you done any experiment to show whether you really need all the variants to be in the vaccine, or maybe one variant can produce broadly neutralizing antibodies?
Well, I think
because the variants, will keep evolving.
Right. Well, I think number one, I think there is some evidence, as you see with the Wuhan and the shots out there today, that to give protection, certainly you're going to have some hit or lower protection with some of the variants than others. But the beautiful thing here is, just like a flu shot contains 4 different proteins, theoretically, because of the large volumes, low cost, we could blend different variants if we want and make it trivalent or tetravalent. But there's some evidence you don't need to do that. But let's see, there's the jury is still out. Let's put it that way for everybody's vaccine, and we're no different. The difference here is we have the ability to keep the cost down and the volume up if we want to make mixtures.
Great. So another question here. Are you able to indicate which countries in Africa are being considered for clinical trials?
I'm not able to do it because of confidentiality, but of course I'm able to, but I can't do it at the moment on here. So under NDA, of course. And by the way, if you're listening and you have a clinical trial opportunity that you want to bring to us, we'll be happy to talk to you.
Okay. Are you planning to do phase 1 clinical trials in parallel? You want to finish the phase 1 in Germany, and then you do subsequent trials, phases 2 and 3 in Africa?
Yes, we're definitely considering that and looking at that and looking for the collaborators to do that with, not just in Africa, but also in different parts of the world.
Okay, great. Yes, any other? We still have time. We still have 10 minutes. Are there, Mark, are there other things that you want the audience to know about?
Well, I think the most important points we've gone over. I think the ability to produce more for less, faster, to rapidly modify the cell line to produce the antigen of interest to the party. Just for example, in South Africa, whether you want the South African variant, we already have it. So we would have to obviously run trials with it. If I go back to the slide, again, I want to just drive the point home. People look at mRNA as it's the flavor of the month. It's a new thing. But to be honest with you, we can go just virtually almost as fast because we can take the gene out, put a gene in, change nothing else, right? So from a regulatory standpoint, the cell line, all the genetics are the same. We just take a different sequence. So you take the RBD with the same exact sort of structure from amino acids X to Y, but with the new variant. And so quickly you now have, whether it's 1 or 2 copies, to your point, if we wanted to, we could put another copy here. You could have 3 or 4 in the same strain, but we don't think that's the right way. We think the right way to do it is make pure components and then blend them together if you want to, but we can do it very fast and very quickly. So I think the key here is we can do it within weeks of what mRNA can do in terms of loading up a sequence, getting a stable cell line. But the advantage is once you have that cell line, which is very rapid in our case as well, you can put it in a E. coli fermenter of virtually any size you want and make up years with the production capacity for the few weeks you might lose on the upside. And then the other point that people don't realize is the purification today. Because you can purify these proteins using a C-tag with only 4 amino acids, where in the past it took you months and months and months to come up with purification schemes. We can now do it within weeks. So that bottleneck in the past of a recombinant protein system doesn't exist any longer, because you can take and purify a protein with a C-tag very quickly and very rapidly. In this case, we did it here without even a C-tag. So you can get into your clinical trials, and whether you want in phase 3 or as a commercial product, you're at least through phase 1 and 2 with a tag. You can even use it in some cases. I think it's only 4 amino acids, so it's immunologically inert or neutral. So there's a variety and flexibility, the robustness, the versatility, the productivity. And again, keep it simple. People are talking about mRNA and bringing in manufacturing mRNA. It's going to be difficult and costly. And I think it's a match made in heaven, so to speak, for the African continent, because you can produce a lot for less and get it into the arms of human beings that need help. And again, it's not just for COVID. It's for all the different varieties of things that your continent and the world needs more affordably. Because in the end, it all comes down to the same message, right? Yes. How do we get healthcare and make it more accessible and affordable to people all over the planet?
Yes. I have another question for you here. Is the product stable at fridge temperature? That is 4 to 8 degrees.
Yeah, let me just stop sharing because otherwise I got stuck. Okay, so we're testing that, but it looks like that. just like a normal recombinant protein that'll be very stable at room temperature. We've also done some trials and experiments where we can actually make it in a powder form. And so that looks very successful as well, which would make it stable for probably years, if not, longer, and be transported without refrigeration. But that'll add cost to the equation and whether somebody wants to do that or not. But it looks like from our initial experiments that it will be like your 2 to 8 degrees Celsius refrigerated normal conditions, unlike some of the mRNA vaccines.
Great. So I have another question. So regardless of which African countries the vaccines may be made in, can you expand upon ideally what the country needs to invest in terms of GMP facilities, and what would be the timeline for Dyadic to be able to start making a vaccine and a booster vaccine in Africa if the countries can make available CGMP assets?
Well, it's the clinical trials is the time point. It's not us. We could transfer the technology within a few months. You can run a CGMP batch based on the CGMP production run we just ran, right? So it's getting whether it's a new variant or not, the product through clinical trials. That's the timeline. So as fast as your regulatory agencies will let you go through the clinical trials is how fast we can operate together. It won't be the delay of the manufacturing, because we could even produce, let's say, fill finish in Europe and ship it for clinical trials while you're getting your CGMP facility up and running.
Mm-hmm. Great. Any other question? Well, thank you very much, Mark, for this wonderful presentation. We will take your closing remarks.
Yeah, so my closing remarks are the same as my opening remarks. We're here to help. We're here to provide access. We're here to provide knowledge, expertise, to tech transfer the technology with the right parties for the right reasons. And the right reasons in this case are to help not just the African continent, but a global population stop the spread of COVID-19 and the variants of concern as they continue to emerge. And in order to do that, I think we need more robust, versatile, productive, affordable technology. It's simple and easy. And I think we, after 25 years and leveraging the industrial experience in the last 5 years, modifying it in the zoonosis anticipation preparative ZAPI group and the Israeli IIBR, we're ready to help, and we're here, and we have the tools to help eradicate this horrific disease, and just as importantly, be ready for tomorrow's pandemic, and then traditional vaccines and drugs to help people have accessible, affordable health care. And we're open for business.
Great. Indeed, you're open for business, and I'm sure some African countries or African manufacturers will be interested in your platform, and through AVMI Secretariat, we will let you know. Thank you very much, and we look forward to doing business with you.
Thank you, and we look forward.
Thank everyone for participating. And please, if you're interested in the technology again, feel free to contact the Secretariat, and then the Secretariat will put you in touch with Dyadic. Thank you very much. Bye.
Great. Thank you.